everyone. So for today, MedEureka, one of the main events of UPPSA every year, it is composed of lecture sessions with invited experts from various fields of biomedical and clinical research. This event aims to bring science to the people through seminars and discussion forums. This year, in the midst of the health crisis that the world is facing today, while in pursuit of science, MedEureka goes online for the first time as MedEureka Specials, Cells and All, the value of molecular biology in synthetic biology and disease. So once again, good evening, ladies and gentlemen. I'm Karen from the University of the Philippines Physician Scientist Association, and I'll be your host for tonight. Once again, just for our house rules, please don't forget to mute your microphones. And later in the open forum, you may place your questions through our Slido link with the code 633105. I will be reading your questions for our speaker later. You may upvote the questions that also speak to your curiosity. Again, the Slido code is 633105. Now to start our event, let's welcome Ms. Leslie Cando, President of the UPPSA, for her opening remarks and to introduce us to our MedEureka speaker for tonight.
Hello. Okay. So, good evening, everyone. Welcome to the MedEureka. This is actually our first MedEureka for this year, and as mentioned, it is also our first online MedEureka. So, as introduced a while ago, MedEureka has been a regular event of the UPPSA where scientists share their knowledge to us. So, PSA believes that science is meant to be shared to the people, and this is one of the ways that we can help bridge this gap. And the important the importance of this has not been any more evident um, during these times. So thank you for taking your time to join us in this session tonight. I also thank the wonderful team who organized this event. So I hope we will all learn a lot tonight with the two topics that our speaker will be discussing tonight. So without further ado, I will now introduce our speaker. So our speaker is a research fellow, collaborator from Massachusetts General Hospital, Harvard Medical School. She's a visiting scientist of the Philippine Council for Health Research and Development. Her research interest includes the development of novel diagnostic and therapeutics against acquired resistance in cancer and control of infectious diseases with a goal to establish translational research in the Southeast Asia. So to talk to us on the design, chemical synthesis, and biological evaluation of water-soluble molecules that protect cells from anoikis, which is a form of programmed cell death. Let us all welcome Dr. Heidi F. Cabanas.
Yeah, I guess good evening to everyone. This is, I'm still not used to, webinars and Zoom presentations, but yeah, thank you to UPPSA for inviting me to this event. And it's an honor to be presenting to um, young, bright aspiring physicians and scientists. And I know we also have faculty and other researchers and in our audience. I'm happy that you're here as well to listen to my talk. And first, I want to say kudos to the organizing team and the communications team of UPPSA for really making my talk sound appealing to the general audience. Honestly, it was only last night that I saw your social media post, and you did a great job in explaining, what my talk is all about. And yeah, without further ado, I'll be talking about cells and all the value of molecular biology in um, synthetic biology. Uh, and this is actually in relation to two of the, two of my projects, which is synthetic fibronectin mimic and their use in cell transplantation and the chemical approach to disease-linked antigens. Oops. But before I start, I want to, uh, I guess, describe to you my biomedical research journey. And the reason for this is I know that you guys are physician scientists, and a lot of you would want to do research beside doing clinical work. And I'm very fortunate that I've had this broad experience in biomedical research. So this graph is not to show you how old or the length of experience that I have, but to tell you that it's really hard for me to label myself to a particular field. And at the same time, I also want to acknowledge that my first exposure to biomedical research is in the same university where you are all right now, and where I also got my undergraduate and master's in biochemistry, and that is in University of the Philippines, Manila, in particular in the Department of Biochemistry and Molecular Biology. During that time, I did my research work under my mentor, Dr. Kawili. So what we did is we designed and characterized peptide that binds to Staphylococcus aureus that represents an antigen from Staphylococcus aureus and binds antibody against it. So we design antipeptide antibody. Their goal really is to have to develop a diagnostic tool against Staph aureus. So we injected and we immunized mice, and then we found, and it resulted to antibody titer against the peptide that we injected. So that was a proof of concept. So and I'm very proud of that work, and again, my official exposure to biomedical research. And then from then on, I had further training in chemical biology for my PhD and also for my postdoc. And eventually, I went into cell biology and translational research. And for tonight's talk, I'll be talking about my research on chemical biology. So these are very similar field and with very similar name with that of biochemistry, as you could see, biochem and chem bio. But the difference is that chemical biology is broadly defined as the application of chemistry to study molecular events in biological systems. So I don't know if that, made it easier for you or for me to understand the difference between chemical biology and biochemistry. So we use a lot of techniques in biochemistry in order to interrogate, biological systems. So what I'm showing you here is a picture of what a chemical biologist usually do. So if you heard of high throughput chemical screen, so those are what some of what chemical biologists do. And for example, if you have a project, now if you're studying proteins, and if you want to know how it folds and its function, you're probably doing a project in biochemistry. But if you add chemical to a cell or to any biomolecule, and you want to see, its different pathways involved, so you're doing a project in chemical biology. So yeah, and chemical biology also use a lot of medicinal chemistry. So in order to have their starting compounds and also to elucidate the mechanism, we also do a lot of techniques from cell biology. And before I proceed to my actual talk, I want to show you this pipeline of the techniques that we do in chemical biology laboratory. So this would also help you guys to understand my two talks later. And this is what we usually do. So we first start with the design and synthesis of compounds. This could be through in silico, maybe by software. You wanted to design a compound that binds against a particular target. So, but or you may want to extract compounds from natural resources like bacteria or microorganisms or plants. But your goal here is to have your actual compounds. Then from there, since the step is really to screen for a chemical hit. So you make your chemical library. So these are different platforms where you put your chemicals. You have, it could be in liquid form. So you have a 96-well plate where each of the compounds are there. So you have, you know, the compounds a priori before you screen them. Or you could also have compounds that are embedded in solid phase support, such as beads or metals. So mostly these compounds, you don't know a priori the structure of this compound. So you need to have a step in order to identify them. So the next step is once you have your chemical libraries, you're now ready to screen. So there are two types of screening. One is target screening. In this type of screen, you already know where the compound will bind. So it could be protein, antibodies, or specific macromolecule. So the next type of screen is phenotypic screen. And here you're looking for a particular outcome or result. Like, for example, you add a compound, you wanted to look for a compound that would be cytotoxic to a particular cell, example, cancer cell, but it's not cytotoxic to a normal cell. So you're looking for a specific phenotype, but you don't know where the compound will bind. Then from then on, you get to have a hit or multiple hits. So after that, you need to identify your hit. If it's, again, a solid support, you want to elucidate the structure of that compound. We usually utilize a mass spectrometry or NMR to identify the compound. And from there, you want to further characterize the compound, like you want to see how tight it binds to a particular target. And if you don't know the target yet, you will need to identify the target. So one way to do that is you use your compound as your affinity link. So you immobilize it into a solid support, and then you add your either cell or proteome, and then from there, its target molecule will bind to the compound, and then you do immunoprecipitation, and then isolate it, and then identify it again through mass spectrometry. And finally, you want to know the mechanism of action of your compound, that when you add it or when it binds to its target. So what signaling pathways are affected? So here you utilize Western blot, microarray, or sequencing, different biochemical techniques. So usually chemical biology ends there once you know the mechanism of action. And if you're lucky, you could further characterize your compound based on its in vivo activity by using mice, primates, or other in vivo models. So from there, I want to proceed officially to my talk. So the first talk is, I'm going to talk about this as a synthetic fibronectin mimic, which has an anoikis inhibiting ability and finds its application to cell transplantation. So this study was done during my PhD in Oisugi group in Kyoto University. So the lab has usually conducts phenotypic screen, which I explained earlier. So our lab has a chemical library which is composed of 70,000 plus synthetic molecules. So these are in here you see a 96-well plate. So each well would have a compound in it. So most of these libraries are commercially available. So, but some of them, if we have medicinal chemists in the lab, they would be synthesizing more chemicals. Even the side products or byproducts of their synthesis, we would use that and include those in our chemical library. So we use these libraries to screen for a particular phenotype. And using those libraries, I'm just showing you here that some of the compounds that were discovered using those libraries, which finds with their specific binding target. Now, what I'm going to talk to you is one research effort in the lab, which is that of cell therapy. So as you all know, cell therapy, it's a very exciting field, and it has various applications. And the lab indeed has discovered several molecules which could be utilized for cell therapy. Some of these are adhesamine, which is used for cell culture, for cell expansion, and some compounds are able to differentiate your stem cell into differentiated cells. But when you get into your pool of cells that are differentiated and undifferentiated, you need to have a way to select for them. So we also discovered and developed compounds to select for just a differentiated cell, which would be used for actual cell transplantation. And the compound that I'm going to describe tonight is compounds that would be useful for actual transplantation and integration. Now, although cell-based therapy is very useful and there's so much research effort done to it, its application remains, or its clinical application remains limited. And this is because it has a major roadblock, which is most of the cells that are transplanted die and they don't get engrafted to the recipient tissue because most of them don't survive and they undergo apoptosis. In particular, cells that are used for transplantation undergo a specific type of apoptosis called anoikis. So anoikis is from the Greek word for homelessness, which is detachment-induced apoptosis. So when a normal cell, cells attach to its substrate via the extracellular matrix, and one of protein components of the extracellular matrix is that of fibronectin. So when you transplant cells, so they lose their signals, extracellular matrix, and then they detached and produce more apoptotic signals and undergo cell death called anoiky. So this, when they float, they die, and you call it anoikis. So in order to design an anoikis inhibiting molecules, so we learned that the extracellular matrix mediates its binding to a cell via these two very important receptors, that of integrin and the heparan sulfate containing syndecan. So if the cell binds to extracellular matrix via these two receptors, so you reduce anoikis or promote a cell survival, thus you improve engraftment efficiency. So shown on this side is a stick model of fibronectin protein, which has its heparin binding domain and the integrin binding domain containing the RGD sequence. So in order to design this anoikis inhibiting molecule or your fibronectin mimic, so we need to have an integrin binding module and heparan sulfate binding module, which is contained in syndecan receptor. So for integrin binding module, I utilized the well-known RGD peptide. This is a tripeptide that's, again, well-known to bind to integrin domain. And for heparan sulfate binding module, I utilized adhesamine. So I mentioned this earlier. So adhesamine was discovered in our laboratory by one of the graduate students there. So it was discovered actually serendipitously. So what happened is that she was working on a different project, and then she observed that in one of the wells, she observed that most of the cells are attached or they increased in cell number. And when she applied that to another type of cell, Jurkat cell, which was a floating cells, they also attach to the bottom of the plate. So, and what's responsible for that is this dumbbell-shaped molecule, which we call adhesamine. So moreover, what's interesting here is that by inducing attachment, adhesamine actually also induces normal cell adhesion. So unlike that of other synthetic compound like polylysine, so adhesamine induces normal cell adhesion. By take a look at actin and focal adhesion kinase, you see that they you see their normal morphology. And when we also checked where the compound was located, we see that they're located also at the cell surface receptor. So And when we further characterize the activity of this compound, we saw that actually the adhesamine molecule induced clustering of heparan sulfate. So you'll see here clustering of syndecan at the cell surface receptor with increasing amount of adhesamine. And to design that, what I did is I covalently linked adhesamine to the RGD peptide. Thus, you have a bifunctional molecule. So furthermore, I also synthesize other, compounds. So I'm just showing here so I designate as a structure or a cartoon to represent specific compounds. So this is just for the peptide, for the dipeptide, for adhesamine, the dumbbell-shaped molecule, adhesamine with RGDs. So from here, I characterize the compound and check its binding interaction first. So with that, I used isothermal titration calorimetry. You probably are familiar with this when you did your biochemistry laboratory courses. So you look for the thermodynamic parameters. So in here, you want to see the interaction between two biomolecule. So just to let you know that the result of this is that even though I changed adhesamine, it didn't affect its binding to its original target, which is heparin. But moreover, by adding the RGD and the serine amino acid, you also have a better solubility. So when I also check the cell viability, this is just to know its effect on cell survival. This is a very straightforward assay, but the novelty here is that we developed this Anoikis condition wherein we use round bottom, non-coated plate, or we just use a tube to induce floating of the cells, to induce cell death. However, when you add a compound, you'll see here that even after 3 days, so most of the cells are alive. And when we compete the compound with that of just adhesamine or the RGD peptide, so I mentioned earlier, we already know where these two compounds bind. So when we added them to the with the adhesamine di-RGDs, you see that the number of cells that survive get reduced. So meaning that they compete to the same binding receptors. So next is to further characterize cell death and survival. So I checked for expression of annexine and PI stain just to make sure that these are not necrotic cells, or check for apoptotic cells. So you'll see here that there's less dying cells for those that were incubated with a compound. And this cells actually are in their GAP2 or M state, meaning they are living and actively dividing. And when I recollected those floating cells and reculture them, you'll see here that this is a crystal violet stain. You see that when I reculture them and then let them grow for a week, you see more cells that are alive. So next, I checked for its mechanism of action, so like what signaling pathways are affected by this compound in order for it to induce cell survival of those, even though they are in a floating condition. So here's a figure that shows you the different proteins involved in apoptosis and survival. So I just check for proteins that are well known to be involved in apoptosis, and that is caspase and BCL-XL, and proteins that are known to be involved in attachment. So I check for the PI3-AKT pathway, the FAK pathway, and the MAP kinase pathway. So I'll be showing you the here first. It's a chemiluminescent assay. So I just check for the caspase, production. You see reduced caspase production for cells incubated with another caspase. So I also checked for this anti-apoptotic protein BCL-XL. You see that there's increased production of this compound because it is anti-apoptosis. Oh, sorry, of this protein. So next, I went to elucidate the mechanism. I went to look at different survival signaling proteins. For this, I use Western blot. So I first checked for AKT and ERG. So indeed, they are the phospho-AKT is upregulated or activated, but not that of the ERK pathway. So I checked more upstream of phospho-AKT of AKT pathway, and indeed I saw upregulation of phospho FAK and not that of PI3K. And oops. Oh, sorry, I didn't. But when I checked for I need to correct that. So when I checked for FAK and PI3K, I did not see significant increase. But when I checked for RAC or CDC, which also upstream of AKT, so I see significant upregulation of or significant phosphorylation of RAC1. And interestingly, the RAC1 is actually activated by integrin. Moreover, when I check for phosphokinase C, it's also gets phosphorylated, which is actually initiated by a syndecan. So what you see here is that integrin and syndecan activates the syndecan activates phosphokinase C, and integrin activates the RAC, which both converge to AKT pathway, leading to cell survival or anoikis inhibition. So furthermore, I also checked integrin and syndecan clustering for this compound. So what you could see here is there's co-clustering of integrin and syndecan for cells that are incubated with the compound. So here's just a Pearson correlation coefficient plot, shows you that they are indeed co-clustered. I also check what type of which integrin binding integrin module gets upregulated, and it's a beta 1 domain of integrin. So next, I look at overall pathways. So in here, I did microarray. So in microarray, so in order to look at the more broad gene expression of the cells that were incubated with the compound. So in here, you extract the RNA and then RT-PCR that to make your cDNA and allow it to, this is a hybridization technique. And then you have a plate which has probe that corresponds to different genes. So in here, there's around like 9,000 probes that represent around 6,000 genes. So as you could see, I would be able to look at like thousands of genes here and not just few genes as compared to the Western blot that I did. So in here, you'll see here, here's a gene scatter plot showing you that there's high correlation of the genes that are enriched for when a cell is exposed to fibronectin, and also when a cell is exposed to our compound. Here's also another Pearson correlation coefficient plot showing you that there's 0.99 similarity for the genes that were expressed for fibronectin activated cells, as well as that from our compounds. So here's a gene ontology analysis. So you guys are, if you're into data science, so what you do here is you have your list of genes and you put it into a database, and that database would group your genes into specific pathways. In here, I look at reactome. So we concluded here is that it's that most of the genes that are involved in integrin cell surface interactions are upregulated. And even for the cellular components, the genes that are involved in extracellular matrix are upregulated as well. And similarly, those in different biological process involved in ECM. So it's the same for the adhesamine mono RGDS. So lastly, I checked for the in vivo activity of this compound. So we used two models to check if they could be utilized for cell transplantation. The first one is we use it in a diabetic mouse model, which, as you know, have a difficulty in wound healing. So in here, so we injected bone marrow derived cells along with the compound. So what you see here is that for after 3 days, so the mice that have no treatment, you see the wound is still enlarged and not healing. This is the mouse with just the cell injected with just the cell only, and then the mouse with the cell plus the compound. You see that the bone marrow cells are getting engrafted, and then they started to heal. And the next model that we used is a rabbit model, which has a bullous keratopathy. It's a corneal endothelial disease in which the cornea, lose its transparency. And usually the treatment for this is you do entire corneal transplant. So what we did is you mimic bullous keratopathy in a rabbit model. And then we injected a cultured corneal endothelial cells plus the compound into the rabbit eye. So it's the same rabbit with and without the compound. So what you see here, you have the no treatment, which is the cell only, and with the cell plus the compound. Oops. You see that there's obviously for no treatment, you don't see any cells engrafted. And then with the cell only, you see few cells, but with the compound, you see most of the cells got engrafted. And even the corneal transparency is a lot better for that, which is the cell only. And you could also see that there's a normal cell morphology even at day 14 as compared to that of cell only. So these results are very promising since you see this in your in vivo models. So finally, we have proof of concept that we could design a bifunctional molecule, synthetic bifunctional molecule that binds to these two receptors, integrin and heparan sulfate containing syndecan, which are known receptors that involve signaling pathways that reduce anoikis and improve engraftment efficiency. And just to show you this, here's a video that shows you the activity of the compound. So you see here, there's a cell membrane with the receptor. So this is integrin, and then your heparan sulfate containing syndecan. So if they don't cluster, you see death of the cell. And then they float, they undergo anoikis. But if you add the compound, it induces clustering of the syndecan and integrin. So by induction of clustering, you activate downstream signaling pathway, and your cells, although they are floating, they are still alive, which you could use for cell transplantation. And of course, that was just a proof of concept. So the future work of this study is that you wanted to design a better compound and then expand its clinical application. And with that, for this first talk, I wanted to thank my previous lab and my mentor, Professor Oyusugi, my previous lab members. And I think I want to especially mention Dr. Punzalan, actually Dr. Enriquez now, she's probably in the audience, and also our collaborators. And my funding source, I was able to do that because of scholarship from the Japanese government and from the Institute for Cell Material Science from Kyoto University, which is a world premier institute, and our supplier of chemicals, which also funded our research, Nagase Pharmaceutical Company. And I want to end this talk with first talk with this quote from Jules Verne, which is the father of science fiction, which he said that science is made up of mistakes, but they are mistakes which it is useful to make because they lead us little by little to the truth. So yeah, that's my first part of the talk.
So thank you very much, Dr. Cabanos. I think it was a really high-yield discussion, and I can personally see how these fibronectin synthetic mimics can be used in clinical practice, especially in cell transplantation. So at this point, we will have a 15-minute open forum for the first talk. You may enter your questions through Slido. Go to slido.com and enter the code shown on the screen, 633105. And you may ask questions in the Zoom chat message to the MedEureka Q&A. So if you would also like to speak up, please use the raise hand function in Zoom. So for the questions, um, so the first question is, thank you for your talk, Ma'am Cabanos. What do you think is the significance of these studies on anoikis regarding the prevalent diseases in the Philippines? Thank you.
Um, I guess it's I so for this anoikis inhibiting molecule, are the main application is in cell transplantation, right? Because it promotes cell survival, it inhibits cell death. So I think that's its main application. So if there are any diseases in the Philippines that you would need cell transplantation, I think that's where you'll find its application clinically. Right now, I know that the prevailing diseases in the Philippines, I don't know if they require cell transplantation. These are cardiovascular diseases, right? And some pulmonary diseases. So I guess you could limit its clinical application to, for example, the diabetic model or those that require bone marrow transplants. So, but then again, so we've only done this in animals. So we haven't done clinical trial for this compound. So, but that's its clinical application. But as a cell culture agent, I think we could utilize this compound, and we could also use this as probe. If you're studying, integrine, if you're studying syndecan, so you could use this as probe by if you would add a fluorescent molecule to this compound, then you would be able to visualize this compound and then see where it is on the cell.
Thank you so much, ma'am. So for the next question, why was the single RGD peptide with adhesamine not used in vivo model treatments?
Oh yeah, that's a great question, and I actually get those questions a lot. So the main reason for that is if you're a peptide, for those working on peptide chemistry, you know that peptides are very prone to protease degradation. So we assume that when if we use the mono RGDS, adhesamine mono RGDS, the RGD moiety would get degraded. So by utilizing the di RGDS version, so even the one side of the RGD gets degraded, so you still have one that is left. So actually, those who are doing drug discovery, they would know the limitation of the peptides.
Okay, but for our next question, I would like to ask why the metastatic cancer cells, which have not yet invaded other organs, don't undergo anoikis, even if they leave their organ of origin. Thank you.
That's a great question. Although I'm not an oncologist, so I've done some research on in the transitional lab that I worked on. As you know, cancer cells, it has this array of all of the signaling pathways, right? So I think what happens with one mechanism of cancer cells is that they get addicted to specific oncogene, right? Which, it's always that oncogene is always stimulated, it's always overexpressed, which allows them, to have what is unlimited cell division and unlimited cell survival. So that's why when you design compounds against, cancer cell, you're looking for inhibitors, because a lot of their proteins are, uh, overexpressed.
So for our next question po,
Sorry, I think I wanted to add to that. So that's the for the for that's for the inherent cancer cell ability. I think there's another aspect on that, which is the the other one is the tumor microenvironment. So somehow, in that metastatic lesion, you have a well activated the tumor microenvironment, which promotes their survival. So yeah, thank you. Sorry, that's a very exciting question. So interesting question. Yeah.
okay lang po. So yung next question po natin, would an overexpression of metalloproteases in a patient wanting to undergo cell transplant be a hindrance to proceeding with cell transplantation?
Yeah. Sorry. So metalloprotease protein. So yeah, I'm thinking about the mechanism. So I probably should ask that person why would he think that overexpression of the metalloprotease um, protein be a hindrance to cell transplantation. So I think the good thing about science is that you don't need to give exact answers always. You could always say, I'm not sure. I don't know the answer to that. So yeah.
So yung next question po, I would like to ask po if recombinant fibronectin could have the same application. What advantage would developing synthetic CPDs have over fibronectin? Compounds, I mean, sorry.
Yeah. Well, for you're talking about injecting a pure protein, recombinant protein to an organism. So it's cost wise, it's definitely like more expensive. So, and if you inject a pure protein, there's there's always the problem of I guess eliciting an immune response against that uh, protein. So yeah, but mainly I think it's the what's this it's the cost and the immune reaction. That's why we always I mean pharmaceutical companies would invest more on synthetic compounds.
Thank you, ma'am. So for our next question, if it would resist apoptosis, then could it be potentially a challenge or counter-regulatory to nearby transformed cells?
That's a great question. Yes. So there's always a possibility that could be teratogenic, that it could since you're promoting survival, so it doesn't only uh, act on your engraft on the cells that you're going to engraft, but also maybe it could bind to your recipient tissue that express both integrin and what is integrin and syndecan. So what you need to do, or what we could do, is we could further design, probably add more domain or module to that compound, that's sort of a homing device that would just bind to your specific cell, like for instance to your bone marrow transplanted cells. So that's another study that you need to do, because you're looking for a specific receptor that's just expressed by that cell. And mind you, that's also another field of study. And I think I showed you like some of the compounds earlier on. We have compounds that um, differentially select for differentiated cells versus undifferentiated cells. So I think for stem cell therapy, specifically for IPS, so if you have like your pool of cells that you differentiate, when you transplant them, so not all of them actually are differentiated. So your undifferentiated cell would have the tendency to be, of course, to be cancerous, to be teratogenic. So you need to remove those. So one way is you could design, molecules that are just specific for that and would be cytotoxic to those cells. Sorry, I'm going,
Okay lang
yeah,
po.
it's very exciting, at least for me. Those questions are very good. I don't know if I answered the question. So yeah, you just need to develop, improve the compound to remove its teratogenic activity. Although what's good about this synthetic compounds is that it's again it's subject to proteases. It gets degraded. So another way is you could lessen the concentration of the compound that you add during transplantation. Yeah.
So thank you very much po, ma'am. I think that's all the questions in Slido for the first talk. So before we proceed, let's first have a 5-minute break. Stretch your arms, your back, relax. Now, while you do that, we will have 2 lucky members of the audience that will receive a simple gift from UPPSA. So let's wait for the... So here's a wheel of names. So drawing, so paikutin na natin ang ruleta. So our first winner, Walter Aljan Espiritu. So I think he's here naman. So we will be in contact with you na lang regarding the prize. So for the next winner, so our next winner is Jovenis Mangera. So we will be in contact na lang din regarding the price. So for reminders lang for our house rules, please don't forget to mute our microphones during the talk. And once again, there will be another 15-minute open forum after the talk, and you may place your questions once again through our Slido link with the code 633105. I will be reading your questions for our speaker later, and you may upvote the questions that also speak your curiosity aloud. Again, the Slido code is 633105. So we will be back at 6:58. You may take your CR breaks or stretch muna tayo before we go on and proceed to the next talk. Thank you. So, good evening again. So, for our next talk, Dr. Cabanos will discuss various methods for the monitoring and manipulation of antigen-specific immune responses. So, without further ado, let's give a warm round of virtual applause to Dr. Cabanos.
So yeah, I guess hello again. Or um, yeah, I'm happy to be back after 5 minutes. But if you will allow me, the question about metalloprotease, I can forgive myself for not being able to answer that question because I actually designed during my page. I actually designed another compound that would act as a inhibitor for metalloproteinase. So the question was that how would a person that overexpresses metalloprotease would be affected during, why is it bad during cell transplantation? It's because these proteins are involved in uh, extracellular matrix degradation, right? So uh, if you overexpress these proteins, so you have degradation of your extracellular matrix proteins, which as I described earlier, help in attachment of the cell. So if for your engrafted cells, you don't have a preformed, extracellular matrix proteins there due to your overexpressed uh, metalloprotease proteins. So that's it. I think that's the answer to that question. So let me go to the second part of I hope I was able to answer the question for that person. So again, let me proceed to the second part of my talk, which is, chemical approach to discovering disease-linked antigens. So this research was done during my postdoc at the Scripps, Florida, under Professor Tom Kodadek. So in this chemical biology library, so we, instead of using, libraries that are in liquid or insoluble form, we use libraries that are in solid support, and we do that through combinatorial chemistry. So we call this libraries combinatorial library. And the type of screen that we do here is through target screening. So we already know our target of interest. So in a combinatorial library, it's the difference to the chemical libraries that I've shown earlier at the Oisugi group is that you have more compounds since you're doing it combinatorially. So earlier I showed you the number of compounds were just into 5 digits, but in here you have 100,000s to millions of synthetic compounds which make up your library. So in Tom's lab, in order to make our libraries, we utilize the one bead, one compound combinatorial library. And you usually do this when you synthesize peptides. So if you remember again your biochemistry, when you do solid phase peptide synthesis, you first, deprotect and then wash and then couple with the next amino acids and then deprotect again, and then until you have your peptides and cleave them from the solid support. But in here, instead of using peptide, we use peptoid. So peptoid is an alkylated amine. So it has advantage over that of peptide. For one, it has a wider side chain diversity. So for peptide, you only have your 20 different amino acids. You have 20 different residues. In here, you have hundreds different side chains. So you have a more diverse library, and you could also make these libraries quicker. So in peptide synthesis, you still need to do the deprotection. In here, you don't have that deprotection step. And of course, with that side chain diversity, you have a very diverse chemical space. And another advantage for peptoid is that it's protease resistant. So, which is one of the limitation for peptide if you're designing compounds that you want to be, more stable. And this is also more cell permeable as compared to peptide. But of course, it also has its, disadvantages. One of that is it's very floppy. So there's no steric hindrance as opposed to that of peptide. So it would affect its specificity. So that's why the hits that you get using this combinatorial peptide library. So you still need like a step to, I guess, all primary screen hits. You still need to optimize those screen hits. So let me just describe to you how we make this combinatorial library using peptoids. So it has two simple steps. You just acylate your beads with bromoacetic acid, and then you displace that with available amines. So those were just like the two steps, and then you use that to make your split and pool synthesis. So for example, you have your first bead, and then you activate that using bromoacetic acid. Now this is now ready for amine displacement. And then in this example, I use 3 different amines. So after you split it, and then you add the amines, and then now you have a diversity of 3, right? And then you pull it again. And then after pulling that, so you acylate or activate it again, then you split it, and then it's ready for amination. So in here. So I split it into 9, and then I add again another different amine. So you have a diversity of 3 times 9. So now you have 27 different compounds. So of course you do that again. You split and then you pull it again until you make a what is a more diverse and bigger libraries. So in a peptide library, you have a diverse chemical space. So we wanted to utilize this diverse chemical space in order to look for disease-specific antigen or adaptive immune response. So the utilization of this is mainly for immunodiagnostics. As you all know, our antibody repertoire that's found in serum would represent the key adaptive components of the B-cell mediated humoral immunity. And this diversity of antibodies that we have in our blood or serum is shaped by the record of exposure to different antigens. Could be, microbial antigens, self-antigens, or neoantigens. And since some of these, antibodies were raised against specific antigen and specific disease, in this case, the red antibodies here. So these antibodies could be, of course, biomarkers for a particular disease. And this idea is not unique to our lab at Scripps. And as you all know, the Dr. Kawili's lab also utilized this idea to fish out disease-linked antibodies. So in his case, we used peptides. So, but the end goal for this immunodiagnostics really is to use this chemical compounds as high affinity antibody capture agent, that when you immobilize it and then identify that specific antigen. And even though, a lot of labs are doing it, including the one again at Biochem in UP Manila, so there's still a lot of diseases that there's no definitive blood test in which we could utilize this technology. So such as the diseases indicated here. So with this, even though you're utilizing this chemical space, it's really hard to look for disease sera, or disease or antibody that is linked to disease. It's almost like you're looking at the needle in a haystack. So again, so our goal is, once you detect this disease-specific antibodies, then we get to know that disease-specific antigens. And we want to do this blindly, because what most people do is that when they look for native antigens, they usually perform comparative array screening. So in this example, you have an array here where you have your recombinant proteins, peptides, or lipids that are already like known component of an antigen. So what people usually do is that they expose that to a healthy and a diseased sera, and then add a labeling reagent, and then just look for a well that just specific for the diseased sera. So the limitation here is that you're limited to, again, like for recombinant proteins, you're limited to just proteins that doesn't have a post-translational modification. And what if this native antigens have those post-translational modification? And also there's still like a lot of unknown, again, native antigens in the immune repertoire. So again, what we wanted to do is to take advantage of this diverse chemical space in order to search for synthetic and antigen epitopes. So we actually call this antigen epitope surrogates. So our question is that can we find synthetic molecules that occupy the antigen binding site of these antibodies, even if they do not resemble conventional biomolecules that I mentioned earlier, such as proteins, peptides, and lipids. So how do we do this? So again, we start with our library. So for example, here's a chemical library that I synthesized. So, and we perform, instead of a comparative array screen, we perform a subtractive serum screening approach. So what we do is that, so what you see here, here's a, this is what we use for synthesis. The yellow stuff there are actually beads. So on those beads, that's where the chemical compounds are located or were synthesized. So you could use these beads directly for screening. So for example, here I have a 10 to the 5th bead. So that's how much compounds I have. And then I expose that to a healthy sera. And then from that healthy sera, I remove those that are non-binders. So we do this 2 ways. We could either do magnetic screen to remove those beads, or we do it under the microscope. We use a red Q dot and then we manually pick those beads and then discard them. And then just the next step is to use those beads that didn't bind to the healthy sera and then expose them to the diseased sera. So this time, those that, beads that bind to the diseased sera would be our hits. So from there, since we don't know yet the structure of these compounds, we identify them through mass spec, and then we resynthesize them and purify, and then put them in a microarray or flow cytometry, and then re-expose them to another set of sera. So as a proof of, so the next step that we do is that once we have our hits, we, again, I said we either do microarray or flow cytometry, but in this case, we have developed a technique using flow cytometry to do the second step of screening from our hit compounds. So in here, we developed this Luminex technology. If you've heard of if you've seen, a flow cytometry panel of colors, they use beads of different colors. So we utilize that. So in here, we do this in-house. So we color the beads. So we use 2 different dyes in different ratios. So with that, you would have beads that have different colors. And so each of these beads would correspond to a specific compound or specific hit. And then we expose that to our serum, and then we add another antibody containing fluorophore, which would detect the binding of the disease sera to the specific compound. So in here, we would use like 2 lasers, one to detect the specific compound and the other laser to detect the emission of the binding of the antibody. So as a proof of concept, so using that, the subtractive screening and then the Luminex technology. So we found this compound KTD1, which binds to diabetic mouse model. So again, our goal is really to identify the native native antigen. So we use this as a affinity column. So we expose this compound into a lysate. In particular, we use the mice brain lysate for this, and then we communiprecipitate that, and we got this band here. And when we checked it through LC mass spec using proteomics, we found that this band correspond to GAD65 protein, which is a protein that is expressed by pancreatic beta cells. So, and then when we compete that with our compound and the protein, you would see that they bind to the same receptor. And furthermore, these are other compounds that were discovered and binds to specific disease sera. So using our antigen epitope surrogate technology. So I've shown you the KTD1. So we have another, and we found that the antigen is GAD65. We also have another compound that binds to, and we were able to, to fish out peripherin as the antigen. And then this is for chronic lymphocyte leukemia. There's there's a compound that binds to disease sera with Alzheimer's disease, and then which, however, we're not able to identify the native antigen for this one. While for this one, for which is encephalomyelitis, we were able to identify native antigen MOG. But the next slides that I'm going to show you is that, of course, these are all like positive results using this technology. However, science is not always like that. So no matter how big your screening libraries are, sometimes you don't get a hit. So I'm going to show you some of those examples. This is another library that I synthesized. It's actually a much more complicated library. It's composed of 200,000 compounds. And it's much more complicated because you have a cyclic compounds here. So this is the first time that we did that thioether cyclization of peptoids. So the idea here is, if I have a cyclic compounds, I have a more conformationally constrained compounds. That means they're less floppy, they're more stable, and they would probably mimic more the native antigen, since you see that they're more in 3D conformation. So I utilize that compound to set up other serum screens, such as in this one, melanoma. You see that I can show you the structure of the compound here for proprietary purposes. So in here, most of the serum from the deceased sample, from patients that have melanoma, binds to the compound, but those of the healthy samples, very few of them binds to the compound. But yeah, this one still, warrants further studies. We also use that library to screen for patients with Parkinson's disease. Unfortunately, you would see here that despite the complexity of the library, like only one patient, binds to that compound. And not only for immunodiagnostic purposes, we could also utilize this technology maybe for therapeutic purpose. So we could use that to look for inhibitors. So I also set up screen using purified proteins. In here, I expressed RPN10 protein, which is a component of the proteasome. So this has application to multiple myeloma. It's for patients with multiple myeloma, they are very they're very dependent to proteasomal protein. So that's why you have a therapeutic window by inhibiting some of the components of the proteasome. I also set up screen for by expressing antibody from chronic lymphocyte leukemia. So these are all purified proteins. But unfortunately, we did not find any good hits um, from this screen. So the next slide is a improvement of our library, of our one bead, one compound library. So although we could already have a 100,000 compounds using the method that I showed you, but really the limitation of that is when we elucidate the structure of our heat compounds. So if any of you are doing mass spectrometry, if it's very challenging to deconvolute the compound from your beads. So we developed this technology, which is DNA encoded library. So in here, each peptoid, so it has a corresponding DNA nucleotide encoding. So down the line, by DNA encoding, we could elucidate the structure of the compound by next generation sequencing. So you just need to do your PCR and then submit them for next generation sequencing. So in here, I'll just briefly discuss to you how we do the synthesis. So it's similar to what I mentioned. So you have your peptoid synthesis. You again, you acylate with the bromoacetic acid. In here, you have your chloroacetic acid, and then you add your amine. But after you add your amine, you exchange into an aqueous solution and add the nucleotides that correspond to that amine. And then you go back again to your organic solvent, and then you do your peptoid synthesis and then add the corresponding nucleotides. And what's interesting here is that since it's a next gen DNA encoding, we could use a smaller beads. So this is 10 micron beads. And the advantage of this is using the small beads is that we could do this screening through flow cytometry. So in here, this is just a check of the synthesis when we add a fluorescent probe to our DNA encoded library and see if we could fish out that probe. So again, here's our screening strategy for our DNA encoded library. So this is this project is actually funded by the Department of National Defense in the US because of the DNA encoding technology. It's a new chemistry platform. So, and the screening strategy here, again, as I mentioned, it's through flow cytometry. So in here, we use two different colors of antibodies. So we pre-label the sera from the control or the healthy sera, and then we pre-label the case sera with another color. So once you pre-label them, you could mix them now and then add them to your libraries. So when you add them to your libraries, incubate them, and then you process them through flow cytometry. So those beads that do not bind to any antibodies, obviously they will not give any emission in your flow cytometry. And those that binds in the control sera would have this color, which your pre-label color. Those that binds to both the healthy and the control and the case sera would be on this, would have this color, while those that are just specific to the case sera would be on this region. So we will collect this beads and then submit them for next generation sequencing. So this is just how we do the which is deconvolution. So we have a specific region there that we need to do PCR to increase the PCR product for NGS submission. So anyway, as a proof of concept, we use sera from TB patients. Actually, when we started this project, our goal here really is to be able to identify, or to develop, a panel of compounds that could be used for diagnosing TB patients that have latent TB. However, we were only able to identify compounds that differentiate active from healthy patients. So anyway, so we have this library that has 500,000, around 450,000 unique compounds. And we expose that to our sera from TB patients. So you see here. So our hit compounds is hit compound 2A. You see that patients that have active TB have high fluorescence, meaning they have binding to this compound, as compared to that with latent and the control sera. So again, our goal there is really to identify the native antigen and just use that compound to as affinity link. So we exposed that to the TB patients, and then we did immunoprecipitation, and then expose it to, microbial culture filtrate proteins. And you see, we saw this band, and then when we did proteomics, we identified that it's a the AG85 protein expressed by Mycobacterium tuberculosis. And when we use this protein to to identify, other and expose them to TB serum from TB patients, you see there's a high fluorescence of the AG85 protein. And then when we also, use again competitor using our compound, we saw that indeed they they compete for the same binding to the antibody from the disease from the case sera. And when we further validate this, so what we did is we immobilized the AG85 protein to an ELISA plate, so it could be more useful for as an immunodiagnostic tool. And we use samples from another set of TB patients. So you could see that it's very powerful in differentiating active TB patients from that of latent and normal patient. However, you could see here that not all the TB patients would exhibit high fluorescence from or bind to AG85. So it could be that there's other native native antigen where this patient antibodies from patient sera bind. So that's why, so for future study, we will probably make another set of chemical libraries that may represent other antigens. And finally, we also, again, those are like happy results using positive results using these libraries, DNA-encoded libraries. But we also did use those libraries to screen for dengue-specific antigen and Zika-specific antigen. And again, this is just to let you know that it's, again, not all, a positive result. So for this screens, we didn't get any hits. And with that, so I just want to thank my previous lab at Scripps Research Institute, my mentor, Professor Kodadek, and other members, and our collaborators. And again, our funding sources here were from the National Institute of Health, the Department of Defense, and the Bill and Melinda Gates Foundation. But we also did have funding from pharmaceutical companies like OPCO and Takeda Pharmaceuticals. And I want to end this last talk with a quote from Isaac Newton. He said that if I have seen further, it is by standing on the shoulders of giants. And as you could see from the studies that I presented, So a lot of those studies and experiments I was able to do because of the early on studies conducted by other researchers. And at the same time, I also want to give credit to all of my mentors. And right now, I want to recognize my MS Biochemistry mentor, Dr. Salvador Kawili, who really uh, helped me a lot in my journey as a researcher in the biomedical field, and gave me enough training, in order for me to love biomedical research, and also to which is was very supportive of my further trainings abroad. And lastly, I also want to thank and give honor to our essential workers who and frontline workers who have been working hard during this pandemic. I really salute you, and you guys are my hero, considering that I have family members who are affected by COVID-19. And with that, thank you very much. So if you have questions, yeah.
So thank you very much po, Dr. Cabanos. I'm pretty sure we learned a lot regarding the different techniques discussed in identifying disease-linked antigens and compounds. So once again, at this point, we will have a 15-minute open forum for the previous talk. So aside from sending your questions to Slido, you may also, if you want to speak up, you may also use the raise hand function in Zoom. So for our first question in Slido, so ma'am, how did you
Yeah.
differentiate binding of the disease antibody to synthetic compounds versus DNA? Or is the DNA sequence degraded after the synthetic is produced?
Oh no, they're not degraded. So this is big. I forgot to mention. So when we do the this is referring to the DNA encoded library, I assume. So when we do the DNA encoded libraries synthesis, so 99% are the actual compound, and 1% only are the DNA encoding, and that is enough for next generation sequencing. So your antibody would surely the probability that it's going to bind to to your synthetic compound, specifically the peptoid, is higher, of course, because you have the beads is composed 99% of that is covered with peptoid, as opposed to just 1% of your DNA. So yeah, and I also forgot to mention that in order to make sure that it's a real hit, we only look for redundant hits. So what we do is that we have redundant library, meaning we have five, for example, we have 5 copies of the library. So when we do the screen, so if we get a hit and then we check the structure and we only got that once, for us, we don't consider it a true hit. But if we get that structure twice, so that's going to be a real hit. So in that way, we uh, solve the problem of non-specific binding, or and at the same time it non and at the same time we're sure that it didn't bind to either DNA. So it really just it really just bind to the peptoid synthetic moiety. Yeah.
So thank you po, ma'am. So our next question, can these antigen slash epitope surrogates be used po for drug discovery?
Oh, yes, yes. I didn't explain further because we didn't have nice results for that. So I think I did mention what is the multiple myeloma. So yeah, for that, the application for that is yeah, we could design inhibitors so that binds to specific protein target. So in the case for multiple myeloma, so I expressed RPN10, which is a component of proteasome. So I mentioned earlier that which is for um, multiple myeloma. They're multiple myeloma patients, so it's a cancer of the plasma cell. Like you have a unlimited division of your plasma cell, and this is one reason for that is that they are very dependent on proteasome. So you have overexpression of proteasome for these plasma cells, and one component of proteasome is the RPN10 protein. So I express RPN10 protein and then expose that to my chemical libraries, which represent your inhibitors. So, so you're actually looking for a compound that binds to RPN10, which would inhibit its proteasomal activity. So yeah, so now your hit there, or your goal there is a drug, that would inhibit the proteasomal activity and would stop overproduction of abnormal plasma cells, which is applicable for multiple myeloma. And then another therapeutic application for that, for drug discovery, the one that I discussed, is the chronic lymphocyte leukemia. So there's production of abnormal uh, antibody for that. So that's why I expressed the what's this CLL antibody. So I was looking for a inhibitor that binds to that abnormal antibody. So if I find an inhibitor again that would suppress production of that antibody or would degrade that antibody, then you have a drug that could be used for chronic lymphocyte leukemia patients. So I guess the key there is you need to look for a disease that you have a therapeutic window that you could design inhibitors. And yeah, this antigen epitope surrogates could be designed or utilized as, for therapeutic application, such as drug inhibitors.
Thank you po, ma'am. So for our next question, have there been instances of non-specific binding in non-communicable disease-associated antigens and communicable disease-associated antigens?
Has there been non-specific binding for these compounds? Sorry, so I'll just answer it the way I understood the questions. Has there been like non-specific binding of these compounds to could you
Non-communicable
read the
disease.
question again?
So
Sorry.
how sige po. Have there been instances of non-specific binding in non-communicable and communicable disease-associated antigens po?
Non-specific binding of the compounds to antigens found in communicable and non-communicable diseases. Well, I actually don't understand my apologies. It's my comprehension of the question. So, so we're actually looking for compounds. In general, the compounds they would always have non-specific binding. So even for a specific drug, when you design a kinase inhibitor, they actually binds to other kinases, right? That's why for pharmaceutical companies, they do a kinome scan. They would really check for each of the kinase. Does it have binding? So, and definitely and that's already a very like specific compounds, and for this peptoid libraries, again, their problem is that it's a floppy molecule, so it definitely has a non-specific binding, and that's why we further optimize our hit compound. So we have medicinal chemists that would really redesign or what is change this compounds. So yeah, I don't know if I answered that. So yeah, there's like non-specific binding, but you don't want that. That's why you want to design a more specific compound. And yeah, it has application to both communicable and non-communicable diseases. or another way, I guess, to answer that question is that do they cross contaminate or cross over, like the antigen that we find, like for instance in a dengue, uh, are they also could are they also found in, like, I guess, non like cancer patients? So I don't know, again, because we're looking for specific for specific antigen that would uh, differentiate those patients. But these are again, these are usual proteins. The GAD65. They are expressed in both healthy and healthy and a disease sera. It's just that they're just overexpressed. So yeah,
So
that you
for
could. I mean, they're overexpressed that you could fish them out. So yeah.
okay po. So for our next question po, was the method in getting a hit compound ever used in conjunction with in silico methods such as molecular docking simulations?
In our lab, in both our labs, in what's this in Kyoto University and at Scripps, we never did in silico screening. We always start with the, I guess, just our chemistry, and then we look for literatures, like what are known structure of those antigens. But I know there are labs that do in silico screening, but I know that for those labs, mostly they do drug repurposing. So these are already known compounds. So there's a database for those compounds that have the structures, and then they do docking. For the scripts, again, epitope surrogates, uh, our uh, I guess advantage is that we're exploring a new chemical space. So structures that could be patented and could be sold by the lab. So yeah.
Okay po, for our next question, may I ask po in what ways this novel technology differ or if more advantageous from phage display library?
I haven't done phage display library. Uh, I guess for phage display, you do, if I remember. So again, you're for phage display, you're limited to like peptides. You're also limited to antibodies. These are like known. You have known, um, or the structure a priori. So, but again, in the antigens surrogate using these libraries, we're we're exploring a diverse and a newer chemical space. So yeah, and uh, I don't know which is more expensive. I assume phage display is more expensive, and then for phage display, you also need to you need to do the screening platform there is through flow cytometry as well. So with the peptoid, of course we do flow cytometry, but we could also do magnetic screen and microscope screen. But mainly it's the diversity in the chemical space, so which could not be offered by peptide and antibody phage display.
So for clarification po, what is the significance of using two dyes in the tentagel microspheres? Thank you.
Oh, yeah. So the two dyes, it's so specifically we use Pacific blue and Pacific orange dye. So it's to encode the so you'll have different colors. So we use different ratios of this dye So you have different intensity of colors, right? So if you have different, so this different intensity of colors would represent specific compound. So intensity one would be compound one, two, compound two. So, and we use those two dyes because what you want is dyes that have the similar excitation, because we screen through flow cytometry. So we use lasers and detect the emission of that. So you use dyes that have the similar excitation but different emission, so you could differentiate them. So that's the advantage of using two dyes. But if you use, of course, you could use the different colors, right? However, if you use that, then you need different excitations. So in order to identify that, and which you don't want, or you're limited to a very more sophisticated flow cytometry equipment. Yeah. Oh, sorry. And at the same time, for when you choose those dyes, it should be the emission should be far from your third dye, which is the one against the antibody. So yeah, I wish we have a whiteboard where we could.
So for our next question po, hello, Dr. Cabanos. Sorry if I was able to miss it. I just want to clarify what is unique in using peptoids for your study instead of peptides po, and what would be its benefits and limitations po. Thanks po.
Oh yeah, that you're right. I explained that earlier in my talk. So it's a peptide. It's an alkylated what's this amino acid. It's the advantage of that is that it is what's this in terms of the chemical property. I said it's more permeable, and then it's less protease resistant as compared to peptides. And another one is you have more amines, so you're not limited. So that means you have more diversity. So you're not limited to your 20 amino acid residues, but you have hundreds of amines. So just imagine the combination of that, like 100 times 100 times 100. So you have a bigger library as compared to your 20 times 20 times 20, right, of your peptides. Now, again, since it's uh, what's this? The structure is out of the usual, uh, out of the conventional biomolecule. That's why we were hunting for unknown antigens, right? So it has some limitation in its application. But for the peptide, so the disadvantage or limitation of that is, again, based from the structure of these peptoids, they're very floppy. So that means they're less selective. So that's why you need a lot of chemistry. And again, and peptide, it's an natural biomolecule. You would expect that they'll be more specific, and they probably have you'll probably be able to fish out more of the naturally occurring antigen with that, because again you find that in our biological system, and that's what the antigens are made of the peptides, right? So
Okay po. So for our last question, out of curiosity po, what is the proposed function of KHFC1 in melanoma? Thank you.
so yeah, the KFC1 stands for Kodatic Hydifrisco Cabanos compound number one. So the proposed function of that is really for immunodiagnostics, for diagnosis of uh, melanoma patients. So um, usually when you get to it's a what's this? It has high occurrence in the U.S. obviously, but not for not for us Filipinos. And so when they diagnose melanoma, usually it's because you already see the symptoms, right? You have already enlarged high pigmentation in your skin. So what we wanted to do is to, be able to diagnose that, even though you don't see the phenotype or the symptoms yet. So maybe they already have neoantigens in their blood for the melanoma patients that we could detect prior to, unobservable phenotype. So that's what we wanted to do. And and yeah, so using that, KHFC1, so we want to fish out the antigen. And even if we don't get to find an antigen for that, at least for, I mean, just that synthetic compound, we could use that as an diagnostic tool. So, yeah.
So thank you very much po, Dr. Cabanos.
Thank
So
you as well. I really enjoyed the Q&A. I hope it's not just me who's enjoying the Q&A.
I'm pretty sure naman po na yung mga questions naman po na sinend nila nasagot naman po. And it was a very fruitful discussion naman po. So at this point
Great to hear here.
of the program, we would like to express our appreciation to Dr. Cabanos. May we present the certification? So this reads Certificate of Appreciation. This certificate is given to Ma'am Heidi Frisco Cabanos, PhD, for sharing her invaluable knowledge and expertise as the resource speaker during the MedEureka special entitled Cells and All, The Value of Molecular Biology in Synthetic Biology and Disease, given this 16th of April 2021 via Zoom. This is signed by Ms. Leslie Faye Cando, the head of the UPPSA, and by Ms. Carol Ann Baldo, which is the program head of this event. So thank you so much po for sharing with us your research, Dr. Cabanos. May this new knowledge be like a match that might ignite the ever-curious hearts of our aspiring researchers in the audience to venturing into the field of cells and all. So for everyone, this doesn't really end here. We will have another MedEureka session. So when ba to? It takes place next week, April 24, 2021, next Saturday, entitled MedEureka, Coursing Through the Pandemic. This series of sessions tackles topics on basic sciences, clinical approach, local and national infectious disease or COVID-19 response, and towards public health in the context of the COVID-19 pandemic. So if you guys want to join us, shown is the registration link for the said event. So you may sign up at bit.ly MedEureka COVID. And also, it would be really appreciated to hear your thoughts in the discussion with Dr. Cabanos. Please follow the link to our feedback form. Both links for the registration and feedback form will also be provided in the Zoom chat. Certificates will be given to those who accomplish the feedback form. So to conclude our event, may we welcome Dr. Salvador Eugenio Kawili, our UP PSA advisor and UP College of Medicine, Department of Biochemistry and Molecular Biology faculty, for his closing remarks.
Okay. Good evening, everyone. Can you hear me?
Yes,
Hello? Oh, okay. Yes. Good evening. So, I would like to thank everyone who made this event possible. Of course, PSA. I distinctly remember the PSA, the last live PSA event that we had in, I think it was like February last year, when we had a visitor from Germany who was a friend of another of our big public scientist awardees, and uh, it's it's uh, it's heartwarming to know that despite everything we've been through, we're still moving forward. And of course, I'd like to thank uh, Dr. Heidi Cabanos, especially for being our Balik Scientist guest. So we're trying to engage as many Balik scientist guests as we can. And I think that I should say that, I mean, one of the greatest feelings that a faculty member can have is like to actually uh, uh, be a witness to the success of for former students, uh, and even beyond that, to actually have former students be successful and return as collaborators. That's really something that I think is very fulfilling for faculty members, and I hope that at least some of the younger members of the audience would actually eventually go through the academic track and have this very rewarding feeling themselves as well. And I think it gives us hope. No? We've been very hard hit, and we realize actually that we should have been doing things collectively as a country, starting many decades ago, to have a better response to things like the COVID-19 pandemic. But I think to put a positive spin on it, it's a period for us to have this what I would call a reawakening. I think that a lot of us don't realize that we as a country were able to make our own vaccines, for example, make our own anti-sera. And this was happening even before the Second World War. I think the tragedy there is that uh, we lost what we had, and we even forgot about it. So I think one important task for us all, especially the younger uh, generation, is to really, you know, to relearn what was forgotten, to pick up where we left off, and to make it work again and better for this country and its future. So with that, I'd like to end on a positive note that despite all the challenges, there is success, uh, and Dr. Cabanas is a shining example of that. And I think most importantly, especially the Balik scientists, they serve as role models for us of what we can do, or of our potential, what we can realize together. So with that, I'd like to uh, wish everyone the best in all their endeavors, and have a great weekend. Okay. Thank you.
So thank you so much po, Dr. Kawili. So that's it, guys. This is Karen. It was a pleasure to be your host. See you guys next week in the MedEureka Coursing Through the Pandemic. We would like to request to open your cameras for the photo op.
Yeah, good evening po. So we would like to ask for those who can lang naman to open your cameras for a while for a picture taking. We have two panels lang naman. So I'll be taking the screenshot. So tingin lang sa camera and smile for 5 seconds, I think. Okay. I'll be taking the screenshot in a count of 3. This is the first panel. Smile 1, 2, 3. Okay, second panel po. 1, 2, 3. Thank you.
And that ends this MedEureka session. So see you next week. Thank you for attending. Have a great weekend.
Thank you.
Thank you po, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you. Thank you, Sir Badong.
Thank you. Maraming salamat din po sa inyo.
Thank you, sir.
Thank you. Bye.
Thank you po,
See you.
sir.